Breaking the wall of infectious diseases. How genetics provides new tools for understanding innate immunity. Bruce Beutler, University of Texas Southwestern Medical Center, Dallas.
On November the 9th, 1989, I was working in Dallas as assistant professor at UT Southwestern Medical Center.
Thank you. Infection is, without doubt, the strongest selective pressure that ever operated on our species, and in the long run, it brought the immune system into existence as we know it today. A few numbers tell the story very well. In the year 2010, or rather 2000 in the United Kingdom, the average person lived to be about 80 years. That was the median survival, in fact, in our species. In the same year, things were quite different and not nearly as good. In Mozambique, their median survival was about 39 years. But what's really shocking is that if one went back to Victorian times, when England was a developed country by the standard of the times and ruled the waves, then at that point in our history, in the city of Liverpool, median survival was about 10 years, far worse than it is anywhere in the world today. One could say the same of Breslau in 1690. One could go back and look at archaeologic records from Neolithic and Paleolithic times. And what seems clear is that our species, from about 150 years ago back, was living pretty much in the wild state when it came to defending ourselves against infection. And in fact, all of the death that you see, especially the early death, is mostly due to infectious disease. Now, it wasn't any kind of evolution occurring over this short time scale that changed matters. It was our brains that changed matters. It was human ingenuity. It was the recognition, first of all, that microbes were the cause of the problem, then the implementation of sanitation, immunization, antimicrobial drugs. But these things can be carried only so far. There are certain infections against which one can't immunize, of course, and one can't treat all infections with antibiotics. Antibiotics have been with us for about 70 years, and it's not at all clear that they'll be with us for another 70 or 140 years, and so forth. They're undeniably less effective than they once were. So further progress in the fight against infection and even holding our ground might depend on a deeper understanding of immunity itself. It helps to understand how immunity evolved, and we know that where innate immunity is concerned, some of the toolkit that plants and animals use is conserved, and therefore we think that certain molecules and molecular forms for perception of infection and signaling the presence of infection evolved maybe 1 billion years ago or so. These include receptors that have a leucine-rich repeat motif like this. It includes adapter proteins, proteins that detect microbes in the cytosol of plants and animals both. Where animals are concerned, dinoflagellates were thought to have been the origin of subsequent life forms, and for a long time things didn't mineralize well, so we have only a sketchy understanding of the earliest life forms. But then something remarkable happened about 500 to 550 million years ago. This is known as the Cambrian explosion. And over a very brief period in evolutionary time, only about 50 million years, there were mineralizable forms. We see a segmented body plan, the development of limbs, parenchymal organs, eyes, in fact, most of the structures that we would recognize today. And out of the Cambrian explosion, most of the life forms that we're familiar with today evolved. Where the immune system was concerned, something amazing happened at that point too. Prior to this, there was only innate immunity, and it was very sophisticated. Don't think of it as a primitive system, but vertebrates evolved something different. A new kind of cell was born, and this was called the lymphocyte. And lymphocytes were made by various different organs, the typhlozoal in hagfish and lampreys, the thymus, the bone marrow, the bursa fabricius, and lymphocytes were the basis of adaptive immunity. Adaptive immunity is a kind of anticipatory immunity. The amazing thing about lymphocytes is that they can make trillions, literally trillions of different receptors that can recognize almost any foreign molecule that is or that ever could be. This is done clonally. In other words, each lymphocyte has a different receptor-making potential with a different specificity. We also know that adaptive immunity evolved in the presence of a working innate immune system, and it still depends heavily on innate immune cues. And in fact, innate and adaptive immunity operate together with each other, and neither works very well without the other. We can think of the whole immune system as a kind of biological machine distributed across many different types of cells and tissues, depending on many, many different molecules. And it protects us against infection, but not infallibly so, as you've seen. It protects us enough that we can reproduce and we can get by, but we've taken other measures that make us a very different species than we otherwise would be. When we think about almost any kind of machine, it helps to have a list of parts. And the immune system, I can tell you, is much more complicated than an automobile. But if you don't know the parts of the machine, you have a hard time reconstructing it and understanding how it would work. Of course, some parts are more important than other parts. And the LPS receptor was a component of that type. It was something that I began to work on now about 20 years ago. Here in Berlin, that aspect of the story began about 120 years ago. Robert Koch and Richard Pfeiffer discovered that there was a molecular component of gram-negative bacteria that was intrinsically poisonous and made animals look as though they had an infection when this was administered to them. And they called it endotoxin. And it became a very important molecule in medical circles because it was found that endotoxin mediated what we call gram-negative septic shock. If animals or people are infected with gram-negative bacteria, they begin to bleed spontaneously, have organ injury, need to be intubated. All of this was caused by this one type of molecular form characteristic of all gram-negative bacteria. But the receptor for that molecule couldn't be found, and it seemed to me to be the entry point into understanding how we sense infection to begin with. Finding it depended on two particular strains of mice that couldn't respond to LPS. They had spontaneous mutations that made them that way, that made them refractory, and their very existence suggested that there must be a receptor encoded by a single gene. But what was the receptor and how to find it? That's quite a long story. I'll simply tell you that it is possible to track down the cause of a phenotype. This is done by a process called positional cloning, and it was done with these mice, and it took my entire laboratory about 5 years to find the receptor. It turned out to be in a molecule shown in blue here that we call toll-like receptor 4, and it was for that work that a Nobel Prize was ultimately given to my group. Now, it was found in the next year that there were other components of the receptor. You see one of them here in magenta. That's a molecule called MD2, and this molecule may rotate if I click again. We now know that LPS, which is shown in yellow, fits into the MD2, brings this complex together in a certain conformation, and that's where all of endotoxic shock begins. It was found that there were other molecules quite similar to this that would detect other signatures of microbial infection. Gram-positive bacteria, fungi, viruses, all will trigger this family of receptors and elicit a response. But we wanted to find all parts of the whole signaling apparatus, and we needed a faster way of tracking down mutations. First, we needed to create new phenotypes, and we did that with a mutagen in mice, which are a very good model for humans. This mutagen was called ENU, and our goal was to just randomly damage the mouse genome and find instances of bad immune performance, then track down the causes. Phenotypes are genetic traits, and they result from mutations sometimes, but we needed, and we finally developed, a way of tracking down the mutations in real time. We went from 5 years to a situation where now, in about 1 hour, if we see a phenotype, we know its cause. That was our dream for a long time. We thought if only we could look at a family of mice, assuming that this is a pedigree of related individuals carrying 60 or 70 mutations, We wanted some magical pair of glasses that would point to those animals that had phenotypes if it weren't obvious, as I'm showing you here. And not only that, but this magical method would say, this is a mutation in the gene SOX10, and these are the coordinates of the mutation, the protein change, and there's a human homologue, and we even have a structure, and we see where the mutation is within the protein. Now that's all a reality. I'll briefly say how it works. We now sequence all of the mice that have been born to a mutagenized father up front. We know what all the mutations are that can cause phenotype. Then we breed the mice, bringing some of the mutations to homozygosity, and at that point, a computer analyzes the situation and tells us very quickly which mutation is likely to be causative. It's not our goal to hit genes just once, but many, many times, to saturate in depth, and that's what we've begun to do. So how does the computer really tell us when something has happened? This is simply one example. There are many ways that we can be alerted to a mutation, but this is one of the most robust. Here we're looking at a collection of pedigrees which all have mutations in one particular molecule that's under suspicion from earlier screening results. You see by every point here that we're looking actually at hundreds of mutations, but only one of them in a recessive model of inheritance shows a very strong statistical association with a phenotype, in this case, a shortage of CD8 T cells leading to an immune deficiency. And if we want to look at the actual data, we can click on that point and we see that homozygotes for that mutation have fewer CD8 cells than heterozygotes for the mutation or mice from the pedigree that are wild type for the mutation. Remember, there are a lot of pedigrees, and this is all color-coded, and these are control mice. Now, using this system, we kept toll-like receptor signaling under surveillance, and we found a total of 50 mutations in 26 genes that affected toll-like receptor signaling. And I've shown all the mutations in red. These were mutations in the receptors themselves, in chaperones that would bring them where they needed to be, in adapter proteins that would let them signal, in enzymes like kinases and ubiquitin ligases that would carry the signal into the cell, in transcription factors, and in processing enzymes for the cytokine that we measured as the output of the response. This is a fairly complex picture, but you should get the feeling that it's being reduced to real mechanistic understanding, because at this point, many of those molecules have been solved crystallographically. In some cases, I'm being fanciful here, but in other cases, like with the midosome that I showed you earlier, we know how the molecules fit together, we know how ubiquitin chains are attached to some of them, and so on and so forth. We are getting to a state that we really understand how things work. As of a few days ago, we had introduced 74,000 mutations into the mouse genome, hitting about 72% of all genes. And we have a way of monitoring saturation. We can say with confidence that we've destroyed, under observation, 20% of all the genes in the mouse genome and examined the mutations 3 times or more in the homozygous state. And that means that we now know about 1/5 of the parts of the machine. We can anticipate reaching about 50% saturation within the next 3 years. So we're on the road to finding all the parts. Understanding how they fit together is a larger project, and we might expect ultimately to understand as a result who's vulnerable to infection, who isn't. I want to remind you in closing that the immune system, because you have one, poses a certain threat to you as well. You are prone to allergies, some of you. Some of you will develop an autoimmune disease during the course of your lives, and that too we may come to understand. This is a big effort in my lab, which has been going on for the last 4 years, and these are the people who make it work. Thanks very much.